Record this. All right. Hello. Oh, I need to get back a little bit. Welcome to, and I need to turn on the slideshow. This is CJ 352. This is a lecture that may never be used again. It's being recorded in the spring of 2020. I'm Dr. Jason Linville, and this whole lecture is on separation of STRs, but this particular one is going to be a mini lecture that is going to be on specifically the electrophoresis of DNA. All right, so let's go ahead and get this in the current slide. There we go. Okay, so for spring of 2020, and also for every class at this point for 352, before we tackle a new topic, I always like to review where we've been so far. So we've summarized DNA, and we've talked about identifying, way back in the early portion of the course, we talked about identifying blood or semen or saliva, those presumptive tests. And then right before our first exam, we started talking about how to extract DNA. So that's if you have a blood stain, semen stain, chunk of tissue, anything that you think contains cells and DNA in the lab, you begin this process for opening up the cells, sort of separating the cells from the other components, getting it off of the swab, and then opening up the cells and purifying the DNA, getting the DNA extracted away from the proteins that might cause it to degrade, and also at the end trying to concentrate it a little bit. So we wound up right at our first exam. After we physically were able to do the DNA extraction in class, we had this little tube of what looked like water, because it was just sort of a buffer, a water-based solution, and our DNA was dissolved in it because DNA dissolves in water. And what I wanted you guys to know at that time, if I can turn on my little marker down here, if I can find it, is that when we had our tube of DNA, we're thinking that there's sort of like hundreds to thousands of copies of DNA in that tube. Okay, so that's sort of the range. I mean, there could be a little bit more than that. Definitely there could be less if it's poor evidence, but I want you to think of it as terms as sort of having thousands of copies of DNA. Okay, then what we started to learn, and what's going to be on the quiz coming up, is that amplification of DNA, the PCR process, okay? Understanding that if you put a little bit of that DNA in a tube with primers, Taq polymerase, and nucleotides, you're able, by the sequence of the primers, to select the area that you want to amplify and then wind up with, remember when Simone was mowing my yard, wind up with 10 million copies of that allele that you're trying to amplify, okay? So the areas, and then the next thing we went into is, okay, we know how to amplify DNA. What areas are we going to target for forensic science? And throughout the course, we've had those 13 pairs of numbers, and we're beginning to understand, hopefully a little bit more, that the pairs of numbers, the reason they're in pairs is because you get one allele from your mother and one allele from your father, and the numbers are just the number of times those short tandem repeats are repeated. Okay, so we can amplify one area of DNA and we wind up with 10 million copies of two alleles. Again, amplify one area of DNA, wind up with 10 million copies of allele number 1 and allele number 2. And in you, if we're amplifying D5S818 or whatever it is, you may have 10 million copies of an allele that is 14 base pairs long, and your allele number 2 may be 10 million copies of an allele that's 17 base pairs long, okay? I may be, 15 and 16 or whatever. So after amplification, when we put our primers into the tube, when we put our DNA into the tube, when we put our DNTPs and Taq polymerase into the tube and throw them into the thermocycler, now, if we were in class, we would physically be able to hold this tiny tube in front of our face, and it would still look like just a little bit of water in the bottom of the tube, because that's what it is. It's a water-based solution. But in there is not just a small amount of our original DNA extraction dissolved, but we've amplified certain areas of our DNA. And again, we're not amplifying all 3 billion base pairs of the DNA, but the primers are just sitting down and it's amplifying location 1, location 2, location 3. Okay, so we put in, again, a forward and reverse primer for locus 1, for locus 2, for locus 3. So that means we put in 6 primers total to amplify 3 locations of DNA. Okay, the forward and reverse for locus 1 amplified 2 alleles, an allele from your mother, an allele from your father. Same thing with locus 2, and locus 3. So you should have 6 alleles in our tube specifically for what we did in class. So 10 million copies of allele number 1 from D3, 10 million copies of allele number 2 from D3. You have sort of these just 10 million copies of 6 different alleles, and it's all mixed together in that tube. So the next thing that we want to talk about is how we take a small amount of that sample and separate out those 6 alleles from each other. So we get all 10 million of allele 1 to group together, okay, so we can measure them, all allele 2 to group together. And we also want to learn how we measure these, how we physically can tell the difference between, is this allele a 14 repeat or a 15 repeat? Okay, now for us, and this leads us into our first mini lecture, even though the review took a little time to get here, the way we're going to both separate and measure our DNA is through electrophoresis. So essentially, we're going to separate it by racing our DNA, 10 million copies of all 6 of those alleles, race them down a very thin capillary, about the thickness of a sewing needle, and they're going to begin to group together, and the 6 groups are going to be able to separate out. So one group's moving faster than the other 5, okay? And by timing how long it takes the DNA to get from the beginning of the capillary to the end of the capillary, that's going to tell us the size, okay? But as we're going to learn in these next 3 lectures, estimating the size of our alleles, finding out with confidence it's a 14 repeat, it's not as simple as it may seem. It's actually a multi-step process to make sure we're not making any mistakes, okay? In this first mini lecture, we just want to become familiar with, why is it that we can apply a charge on one end of the capillary and another end of the capillary, and DNA actually physically moves through that capillary? So that's what we want to focus on. Okay, classify and describe electrophoresis. Remember, when I say classify, that means, what is the way of separating DNA? And you say, oh, it's electrophoresis. When I say describe, I may say, what is when you apply a charge in negatively charged molecule, or whatever the definition of electrophoresis, you're describing or defining electrophoresis. I want you to be able to do both of those things, okay? Recall, DNA has a negative charge. I wasn't sure how to phrase this, but I'm going to say, what charge does DNA have? Positive, negative, neutral. Just know that DNA is negative. Describe the capillary that DNA travels through during separation. We're going to find that the capillary is actually filled with something called a polymer, and explain why smaller DNA molecules move faster than larger DNA molecules. Number one, no smaller moves faster. And then I also want you to sort of be able to explain it to me why that is. Okay. So with short tandem repeats, these are what we have amplified. These are what we are separating. We're going to separate these STRs by electrophoresis. Okay, so this is a lot of information sort of reviewing, but the key take home here is, how is short tandem repeats? We're not separating them through chromatography or gas chromatography or thin layer chromatography or a liquid extraction. Sometimes we think of that as separation. No, here, our amplified DNA, we're going to separate it by electrophoresis. Okay, what is electrophoresis? Okay, or how does it work for DNA? I mentioned that DNA has a negative charge, so that's one of the things I want you to be able to recall. The reason DNA has a negative charge, which I won't ask you for this, but it helps us as humans to understand, why we're memorizing certain things. If you remember the sugar phosphate backbone, the sides of the ladder, and then the nucleotides, the A, G, C's, T's were the rungs of the ladder, the sugar phosphate along the sides of the ladder, all of those phosphates are like a phosphorus atom, and it's surrounded by oxygen atoms, and one of the oxygen is negative charge. So all of the sides of the ladder have this oxygen on them and a whole bunch of negative charges. So it makes the overall charge of the molecule negative, which allows us, if we put it through some type of electrophoresis, the molecule will move and be able to be separated by electrophoresis, okay? So we're going to see a little mock-up of how we apply this electrical field, but just know that DNA is a negative charge, and you should also know that it migrates towards the positive charge. This is basic magnet knowledge, okay? A negative and a negative repel each other, so if DNA is negative, it will move away from a negative charge, okay? Negative and positive, I can't do that with my fingers, and who cares, you're not watching the webcam. negative and positive, okay, the negative charge molecule will move towards the positive. So if we put a negative charge over here and a positive charge over here and put all of our negative DNA in the capillary, it will move in this direction, okay? All right. So how do you apply? Do you like take little clips and clip it to the end of the capillary? Again, this is sort of a drawing representing our capillary. This is about the thickness of a needle, okay? And it's actually a glass tube, but it's coated with this brown coating, so it actually makes it flexible, okay? You can bend it. So you actually just dunk one end of the capillary into this buffer, okay? And you dunk the other end of the capillary, which actually this end of the capillary eventually goes into an instrument, and the instrument sort of takes over with like a permanent channel of liquid. But anyhow, that channel of liquid, the end of the capillary where the sample goes essentially is in another outlet buffer, which is full of liquid, okay? In addition to the capillary, you stick electrodes in there. So at the beginning buffer where the DNA starts, this inlet buffer over here, you have this little tube, and it's probably, well, the buffer troughs are a little bit bigger, but when we use an old single capillary system, the buffer is about the size of the 1.5 mil tube, the same tube that our DNA extraction is. So it's not a big vial at all. So it's filled up about halfway, 3/4 of the way with buffer, and into that buffer goes your capillary, okay? And also in there goes an electrode to apply the charge. So there's actually, a lot of people get confused, but there's two things sticking in there, okay? So when the electrode gets turned on, it applies a negative charge to this buffer, okay? And the electrode goes into the instrument. It's part of the instrument. Okay, and the other electrode on the other end, okay, here comes the capillary down. The other electrode gets that positive charge. So this is what we were talking about before, where you have a negative over here, a positive over here. So your DNA that's negatively charged is going to want to go from the negative to the positive. Well, the only way for the DNA to get there, okay, which is actually in a separate sample tube, is going to be up through this capillary, okay? So that's sort of the simplified, I know you're saying that's not very simplified, but just know you can apply the charge and force the DNA through the capillary, okay? All right. Now, I had I want you to be able to explain why smaller molecules move faster than larger molecules, okay? And this is sort of where you just have to sort of visualize what's happening inside the capillary. Okay, we're going to blow the capillary up here. So this is the capillary. Oh, great. My mouse button got stuck. Okay, this is the capillary. We are going to, let's try this again. Okay, this is the capillary. Okay, so this is the outside of the capillary, and this is the inside of the capillary, and this is the outside of the capillary. The capillary is actually filled with a gel-like substance, okay? So this is not just a hollow capillary. It's not just filled with buffer. It's filled with like this gelatinous material. So when DNA is going through the capillary, it's not just going through liquid. It's not just floating through there. It's actually sort of forcing its way through this gelatinous material. So the way that you can visualize smaller molecules moving faster is there's just a couple, like over here in this figure, a couple different ways of visualize or thinking about it, okay? So if you think about the polymer as sort of like this fence with different size holes in it, you can see that, and this is actually probably the closest way to the way the gel actually works. It forms polymers and the molecules cross-link, and they have like sort of pores through the molecules. But anyhow, you can envision smaller pieces of DNA sort of are more likely to pass right through the holes in that fence. A larger molecule will sort of bounce off one of the things, and then it may take a couple times, and the molecule has to rearrange itself before it can fit in the hole of that fence. So overall, the smaller molecule can get through that thick gelatinous material faster than the larger molecule, okay? We can also think of it as, you know, down here, what is this called? Ogston sieving. You guys do not need to memorize that term, okay, or use them. It's just sort of a term here. I like to call this the Plinko board, All right, before I get into the Plinko board, I do want to answer Elise's question. Okay, Elise mentioned, and it's a very good comment, especially for those of you here, criminal justice majors have an advantage over the chemistry and biology majors. Because Elise, are you chemistry? Is that right? I get everybody sort of mixed up. I forget. But for the chemistry and biology students, they are cathode is usually, cathode is plus, anode is negative. In certain types of electrophoresis and analysis, that's true, okay? But the cathode and anode, it actually depends on the situation and the way the charge is applied, to where cathode and anode are not always, cathode is not always positive and an anode is not always negative, okay? Even though in a typical situation where you learned it, that's probably was the case. I should have a better explanation as to why it's different in DNA. I think in part because the DNA is the negative molecule and the cathode is where it starts. And so the cathode has to be negative. But just know, you guys do not have to use the term cathode and anode, because I recognize it is very confusing, okay? Just know where it starts, the DNA starts, that is a negative charge, and where the DNA winds up is a positive charge. I'm more concerned that you guys know the overall concept of a negative molecule DNA will move away from a negative charge and towards a positive charge, okay? So the DNA is passing through this gelatinous material, okay? And like I said, the first way this passing through fences, the Plinko board, okay? This looks like a Plinko board, right? And for those of you that aren't familiar with Plinko, it's a game on Price is Right. I know I use Price is Right too much for my analogies, but you take a chip, okay, and you pass it down the Plinko board, which has a bunch of pegs in it, so it looks sort of like this, and it goes the dink, the donk, the dink, the donk, the donk, the dink. Oh, a 5,000? No, it goes in the 0 column at the bottom. Okay, whatever. Sad face. What am I doing? Okay. But the big chip sort of takes a while to bounce between those pegs, but because it's slightly smaller than the space between the pegs, eventually the Plinko chip will wind to the bottom. Well, imagine if you went up to the top of the Plinko board and instead of a chip, you put like a small quarter in there. Okay, well, the quarter would like go swoop, maybe donk one, and just get to the bottom a lot faster because it's smaller. It can navigate between those pegs faster. Okay, you can also think of it as a snake sort of weaving between the pegs or whatever. But anyhow, any way you visualize this, I just want you to understand, because the DNA, and this is sort of the red pen word that I'm looking for there, because the DNA is passing through that thick polymer, the smaller molecules can move faster. So if you say something about the smaller molecules being able to pass through that thick thing, or Plinko board, or holes in fence, okay, just something that you recognize, the smaller the molecule, the quicker it will pass through that gel or that capillary. So this actually shows a single capillary instrument, and this is a little bit confusing because the DNA and the samples actually start here. So this is your negative anode, okay, and then the DNA travels through. This is what the capillary looks like, okay, travels through there, goes through the detector window, and then it continues, this is what I was talking about, in the gel block and goes into the end buffer reservoir. So that's the path of the DNA. Okay, the electrode, which is really hard to see, is actually coming down into the buffer here, and there's an electrode coming out of the instrument here that goes down right next to the capillary and is in the buffer over here. So again, negative, positive, and the negative DNA travels this way through the capillary. Okay. All right. That's a good question. Let's see here. Is there, I should have had a banana in this picture, huh? If you back up, the instrument is probably about 3 feet tall. So it is on a table right here. And this is probably about, I would say, 2 feet across, and then going, it's not quite to the top here. So going up, it's about 3 feet tall. All right. So it's heavy. The reason it's so heavy is there's a huge laser in the instrument that has to zap the DNA and get a fluorescence reading. But so it's sort of like all the weight is in one, so it's sort of awkward to pick up and move because it's like this big machine, but all of the weight is in a very small area of it. Okay. So let's forget about DNA for a while, and let's go to the BRC, the Birmingham Race Course, and let's watch some of these. Oh, wait, those are large dogs for the Birmingham Race Course. Okay, but let's say that with all of the cops aren't going to enforce the laws during a pandemic. I don't even know if the BRC isn't even open. So you have to know somebody who knows somebody to get in there to do some illegal horse race gambling during the coronavirus pandemic. Anyhow, we all know how horse racing works, right? There's a winner. I'm going to ask some questions and I want you guys to throw your answers in the chat box here. Okay. How do we determine what horse wins the horse race? Okay. We measure something and then it's sort of like, you know, one of these, we race and say go, and then at some point we declare one of them the winner. Okay, fastest, okay. All right, and speed, okay, fastest and speed, but some horses can be, you know, fast and then they slow down, and so I guess it was sort of the average speed. So, well, the speed is what? Kilometers per second, right? Here, let's do some fancy math here. So why am I've been in my home and all of a sudden I'm using the metric system for some reason, okay? Or miles per hour, right? Okay, kilometers per second. Okay, well, they all have to race the same distance, right? Kilometers. So what really determines is the time, right? How fast does it take them to get yeah, exactly. How fast does it take? How many seconds does it take to get from the beginning, the starting line, to the finish line, okay? For those of you familiar with track, they do that, or swimming, they do that the same way. They'll have different heats and they'll take the best times, right? So they don't even need to be running at the same time. Sometimes it's not even who gets there first. It's just they look at the time, okay? And in a horse race, the horse that wins is whoever gets there in the lowest amount of time, right? Okay, so we look at these horses and we start racing them, okay? And they're all at the same location at the beginning. They're all at the starting line, okay? But as the race progresses, one horse starts to move faster than the other horses, and eventually at the end of the race, you're going to have one finish a little bit quicker than the other, okay? So it's sort of where somebody's at the finish line. Here, the David Caruso is our judge, I suppose, okay? And he's looking at the finish line, okay? And he sees a horse, and then they stop the clock, and they record a time for horse number 7, okay? Then they record a time for horse number 9, a time for horse number 10, and a time for horse number 12. You compare those times, and you can figure out which one moved faster, right? Okay, horse number 7, 9, 10, and then 12. Okay, so and all of these horses are sort of associated with this time. All right. If we were to draw that out on a chart, okay, this is what that chart would look like, okay? David Caruso is down here, and he's our detector, okay? So he's sort of looking at the finish line. There's his sunglasses, okay? And he's looking at the finish line here to see if a horse is going to cross it. I don't know what I'm doing. There's my horse that sort of has a body, okay? All right. So what's happening here is the horse here is the y-axis, the signal, and this is David Caruso's observations down here. So we can see 1 through 46, which we're not looking at. David Caruso is just sitting there, and the line is flat. There's no horses, okay? And then all of a sudden at 47, now be careful with those drawing edits, please. All of a sudden at 47 seconds, okay, David Caruso says, horse. Okay, we're going to give him eyebrows here. Okay, there we go. Horse, okay, and the signal goes up. He has detected a horse. And then he's like, oh, I no longer see a horse, okay? And then he sits there for another couple seconds and he goes, horse, okay? So you can imagine this diagram is sort of what's happening at the finish line, at the detection line. Nothing is happening. There's no horses. Then all of a sudden there's a horse, okay, there's a horse, and there's a horse, okay? So what we're going to end up looking at with DNA is sort of this flat detection line, and there's going to be peaks every so often that represents when our alleles are coming out. Okay? Yeah, it is beautiful, isn't it? Okay. So we're not racing horses, we're racing DNA fragments, which is great because with horses, some horses have good days, some horses have bad days or run faster than other, track conditions or consideration when you're looking at your racing form. But with DNA, we know that 4 repeats, because they're smaller, are always going to move faster than 6 repeats, okay? Always going to move faster than 7 repeats. So it's very predictable as far as who's going to win the race when we're racing these alleles. Okay. So this is going to end this beginning mini lecture. I'm sorry, I'm trying to think of how I'm going to divide these up. Okay, so what I want you to know from this, again, is know DNA is negative. Okay, know that DNA goes through a capillary. Knows that DNA has a negative charge. And the most important thing, what we spent a little bit too much time on with the horse stuff, is understanding that for DNA, the smaller pieces of DNA are going to move faster through a capillary. Okay, and then we're going to come back to our little example of detecting horses at a finish line. And we're going to do it with DNA. Okay, so I'm going to stop the recording right here very quickly.